The data came back wrong. Not wrong in the way a lab tech makes an error, wrong in the way that requires you to stop, set down your notes, and ask whether you understood the question correctly in the first place. The researcher ran the numbers again. Same result. The dominant haplogroup in the sample was not the one that should have been there. It was not the one that appears in any standard model of how people arrived in the Americas. It was the one you find at the highest frequency in ancient Egypt, in Mesopotamia, in the Levant. The samples came from a population in the mountains of the American Southeast. A people who had been in those highlands, by most accounts, for thousands of years. The numbers did not match the map. Not by a small margin, not by years, by continents. The story, as it has been told for generations, goes like this. The Cherokee are one of the great peoples of the American Southeast, an Iroquoian-speaking nation whose ancestral homeland runs through the ridgelines and river valleys of what is now western North Carolina, eastern Tennessee, northwestern Georgia, and the surrounding territories. They called themselves the Anivier, the principal people, and by every measure of archaeology, linguistics, and oral tradition, they had every right to that name. Scholars have debated exactly how long they have held those mountains. One school of thought, rooted in the Cherokee's own oral history, as recorded by 19th century ethnographers, holds that the tribe migrated southward in ancient times from the Great Lakes region, the ancestral core territory of Iroquoian language peoples. They came down through the cold, fought freezing rains and rival nations, and settled the highlands as their permanent home. Their Iroquoian cousins, the Haudenosaunee, the Erie, the Huron, remained in the north. The Cherokee went south. That is the migration story. A second school of thought, more recent and in some ways more provocative, suggests the opposite, that Proto-Iroquoian language itself originated not in the Great Lakes, but in the Appalachian Highlands, and it was the northern Iroquois who migrated away, not the Cherokee. Anthropologist Thomas R. White, writing in 2007, proposed that the divergence between southern Iroquoian Cherokee and northern Iroquoian languages began approximately 4,000 years ago, right here in the mountains. By this account, the Cherokee were not the ones who moved. They were the ones who stayed. Linguistic analysis supports a very old split. Glottochronology, the study of how languages diverge over time, like a surname that develops regional variants, estimates that Cherokee and its northern Iroquoian relatives separated somewhere around 1,500 to 1,800 years before the Common Era. That is more than 3,000 years ago. Whatever the direction of migration, the Cherokee language was already its own distinct thing while Bronze Age civilizations were rising in the Mediterranean. When the Spanish explorer Hernando de Soto made the first recorded European contact with the Cherokee in May and June of 1540, he found a people with a sophisticated matrilineal clan system identity and inheritance passed through the mother self-governing towns, linked by council and ceremony, and a landscape shaped by generations of maize, bean, and squash cultivation. His chroniclers noted something they could not quite place. These people were different from other southeastern nations in ways that went beyond language. Their customs, their social organization, their ritual life, something about it sat at a slight angle to everything else in the region. In 1775, a trader named James Adair published the first book ever written about American Indians. Adair had lived among the Cherokee and other southeastern tribes for decades, and he made an observation that the academic establishment absorbed, cataloged, and then mostly ignored. The Cherokee, he wrote, displayed a striking resemblance to ancient Semitic practice, clan structures, purification rituals, harvest ceremonies. The patterns he described were specific enough to make him propose earnestly, if unprovably, that the Cherokee might be descended from one of the lost tribes of Israel. Mainstream scholarship did not follow up. The observation was filed under eccentric colonial speculation, and the accepted story moved on. It moved on for almost 2.5 centuries. Here is where it gets complicated. In 2007, a researcher named Donald N. Yates at a company called DNA Consultants began assembling a sample of individuals who claimed matrilineal descent from a Cherokee ancestor specifically, people whose mitochondrial DNA tests had come back with results that didn't fit any expected Native American category. Mitochondrial DNA is the genetic material passed exclusively from mother to child, generation after generation, essentially unchanged. Think of it as a biological surname, not one written in a registry, but one encoded in every cell of your body, passed down through the female line without interruption since before recorded history. The standard Native American mitochondrial haplogroups, the genetic surnames shared by the vast majority of indigenous people across the Americas, are designated by the letters A, B, C, and D. These are the lineages that crossed the land bridge from Siberia into the Americas tens of thousands of years ago. They are the genetic baseline of indigenous America. When researchers test a native population, they expect to find these haplogroups dominating the results. Yates and his team found something else entirely. Phase 1 of the Cherokee DNA project enrolled 52 individuals. When the results were compiled, the dominant haplogroup in the sample was not A, B, C, or D. It was haplogroup T, a lineage whose geographical heartland is Mesopotamia, the ancient river valley between the Tigris and Euphrates, and which reaches its highest global frequency in Egypt at roughly 25% of the population. In the Cherokee sample from Phase 1, haplogroup T appeared at 26.9%. More, haplogroup U, which researchers say has essentially never been documented in Native American populations prior to European contact, appeared in approximately 25% of the sample. Haplogroup J appeared at rates comparable to European populations. And haplogroup X, one of the 5 accepted founding Native American lineages, appeared at levels consistent with a population more like the Druze of the Galilee Hills in northern Israel and Lebanon than any Siberian ancestry group. A 2008 study by researcher Laurent Schlusch identified that Galilee region as a likely center of worldwide haplogroup X diffusion, a finding the Cherokee DNA team considered significant. When Phase 2 added 67 more participants, the pattern held. Haplogroup T remained the most common lineage at 19.4% in Phase 2 and 22.7% across the combined dataset. In 2014, Yates published the findings as a monograph titled Cherokee DNA Studies, Real People Who Proved the Geneticists Wrong. The geographer, Stephen C. Jett, reviewing the work in his 2017 University of Alabama Press book, Ancient Ocean Crossings, called the results Revolutionary DNA Findings, and noted that haplogroups T, X, and J are, in his words, essentially Levantine Eastern Mediterranean in origin. A separate 2006 study published in Molecular Biology and Evolution by Oxford academic, examining 605 individuals across 16 Eastern North American indigenous populations, added a further layer. Male and female genetic histories in this region differ substantially. Among enrolled Cherokee, the Y chromosome haplogroup R1b, a marker associated predominantly with Western Eurasian populations, appeared at roughly 47% of the male sample. Researchers are careful to frame this figure as almost certainly reflecting post-contact European admixture through the male line. But the mitochondrial data, the maternal line, is harder to explain that way. Because the Middle Eastern signatures in the maternal DNA were traced by the research team to female ancestors from the pre-Roman period, before 1492, before Columbus, before contact. Clean, expected, neither. The first theory says this has a simple explanation, and it deserves to be heard on its own terms before anyone reaches for something more dramatic. The Cherokee had extensive contact with European traders, missionaries, and settlers from the 17th century onward, Scots-Irish frontiersmen, Welsh traders, and notably, a significant number of Sephardic Jewish merchants who moved through the southeast in the 1700s and early 1800s. Many of these men formed relationships with Cherokee women. Under the Cherokee matrilineal system, the children of Cherokee mothers were Cherokee, regardless of their father's origin. A Sephardic trader's daughter raised in a Cherokee town, carrying her mother's haplogroup, would have become a Cherokee woman in every meaningful sense, passing her father's Mediterranean mitochondrial lineage forward through the female line for generations. By the time of the 1835 U.S. Census, estimates suggest that roughly a third of the Cherokee population carried some degree of European ancestry. The Trail of Tears in 1838 in 1839 forcibly removed approximately 15,500 Cherokee and killed an estimated 4,000 of them, roughly 1 in 4. The survivors who reconstituted the nation in Indian Territory carried whatever genetic diversity had accumulated through 2 centuries of contact and intermarriage. It is not unreasonable to propose that Mediterranean lineages, arriving through Jewish and Southern European traders, simply became Cherokee lineages over time. This theory is straightforward, scientifically grounded, and carries genuine weight. Its limitation is equally straightforward. The Yates team specifically selected subjects whose results did not fit the post-contact European admixture pattern. Additionally, several of the anomalous Middle Eastern maternal lineages in the sample were traced to female ancestors from periods predating Roman civilization lines that would have needed to arrive in the Americas long before any European sailor crossed the Atlantic. Post-Columbian admixture explains part of the picture. The research team argues it does not explain the shape of the whole. The second theory is more complicated, and the one most researchers approach with significant caution. This explanation holds that some of the anomalous haplogroups in Cherokee maternal DNA reflect pre-Columbian contact, the presence, centuries or millennia before 1492, of Mediterranean or North African populations who left a genetic trace in Eastern North American communities. The centerpiece of this argument is haplogroup X. Unlike haplogroups T and U, X is an accepted founding Native American lineage, one of the 5 haplogroups that crossed into the Americas from the Old World during the initial peopling of the continent. But its global distribution is strange. It appears at elevated levels among Algonquian-speaking peoples, particularly the Ojibwe of the Great Lakes, and it appears at unusually high levels among the Druze of the Galilee Hills in northern Israel and Lebanon, a community that has kept exceptionally detailed genealogical records for centuries, and whose X lineage, the 2008 Schlush study proposed, may represent a very ancient diffusion out of that region. The genetic geography problem compounds when you add haplogroup T. There is no known mechanism, not genetic drift, not founder effect, not parallel mutation that produces T at Egyptian or Mesopotamian frequency in an isolated Appalachian mountain population without a source population. Something carrying T at high frequency had to arrive. The question the data cannot currently answer is when. James Adair's 1,775 observations surface here as a cultural footnote, not as proof of anything, but as a data point that has never been satisfactorily explained. The clan naming, conventions, purification rituals, and harvest ceremonies he documented among the Cherokee bore specific enough resemblance to ancient Hebrew practice that Adair proposed direct descent from the lost tribes of Israel. The DNA does not confirm that hypothesis. Researchers are careful to say the data doesn't confirm it, it only fails to rule it out. The limitation of this theory is substantial. The DNA consultant study enrolled a combined total of approximately 119 subjects across both phases. Participants self-selected based on claiming matrilineal Cherokee descent without verified tribal enrollment, introducing sampling bias that mainstream geneticists have been quick to flag. The pre-Columbian contact hypothesis is taken seriously by a small number of researchers and dismissed or left unaddressed by most. It remains, as of any available study, scientifically unverified at the population level. This is the part the records don't explain, and the third theory is the one that most geneticists and anthropologists are still hesitant to say out loud, because its implications cut deeper than any haplogroup frequency. What if the anomaly in the data is not evidence of something that happened to the Cherokee, but evidence of what the Cherokee always were? Genetic research associated with the History Channel's America Unearthed filming found not one Cherokee genetic profile, but multiple. In some Georgia counties, self-identified Cherokee carried profiles genetically identical to Georgia Creeks, complete with Maya DNA signatures. In one county, the dominant Native American component in so-called Cherokee DNA was Quechua, South American, not North American in origin. An entirely different population signature in a different county again. DNA consultants' autosomal data on enrolled. Federally recognized Cherokee, a sample published in 2016 and expanded in 2018, showed something that sits uncomfortably with every essentialist argument about Native American bloodlines. So-called full-blooded Cherokees registered high levels of European genetic markers and only trace amounts of what the testing framework classifies as Asiatic or standard Native American DNA. Some enrolled members showed almost no Asiatic component at all. This theory proposes that Cherokee has never been a genetic designation. It has always been a political and cultural one, a Confederation identity capacious enough to absorb Creeks, Melungeons, escaped enslaved people, the children of Jewish traders, possibly populations of ancient and uncertain origin, and centuries of internal migration and intermarriage. The bloodline was never a wall. The matrilineal system was not a mechanism for purity. It was a mechanism for continuity, for taking in the stranger and making their children Cherokee. If that is true, then every attempt to identify a single Cherokee genetic signature is not a scientific project. It is a category error. And asking, what is Cherokee DNA, may be precisely the wrong question. Imagine you are standing in the Smoky Mountains in late October of 1838. The air has that specific cold that arrives before the first hard frost, sharp, mineral, carrying the smell of wood smoke and river cane. The leaves are coming down fast. Somewhere below you in the valley, the stockades are full. Soldiers have been moving through the settlements for 5 months now, since May, rounding up families, separating people from their fields, their houses, their burial grounds. You are a woman, a clan mother. You have taken perhaps a dozen people, children mostly, some elders who could not walk the roads fast enough to outrun the soldiers up into the highlands, into the folds of the Smokies where the terrain becomes too difficult for organized military movement. You know these ridges, the way you know the lines of your own hand. You do not know, and you will not ever know, what your maternal lineage contains. Whether the haplogroup passed down through your mother and her mother before that traces to a woman who crossed the Bering Land Bridge 40,000 years ago, or to a woman who lived near the banks of the Nile. That knowledge does not exist yet, will not exist for almost 2 centuries. What you know is the cane and the cold and the names of the people you are trying to keep alive. The descendants of people like you are known today as the Eastern Band of Cherokee Indians. They live at Qualla Boundary in western North Carolina, the same mountains, the same creeks, the same ridgelines. Their ancestors refused to be counted because they refused to be removed. Their DNA, whatever it contains, survived because they ran. The archive was built by the ones who hid. Here is the reframe, and it changes the shape of everything that came before it. The Cherokee are currently the largest federally recognized tribe in the United States, with an enrollment estimated in the hundreds of thousands. Their DNA across every study conducted, every phase of every project produces one of the most genetically diverse profiles of any tribal group in the country. The variation is not accidental. It is not contamination. It is the signature of a people who survived by being more, not less, permeable to the world around them. For nearly 2 centuries, the United States government invested enormous institutional energy in the project of determining who was really Cherokee. The Dawes rolls of the early 1900s assigned blood quantum fractions to tribal members. Enrollment disputes have continued into the present day. The underlying logic that Cherokeeness is a genetic category, measurable by the presence or absence of specific ancestral markers, is the same logic that drove the Indian Removal Act. It is the same logic the Trail of Tears enforced with rifles and winter. The DNA does not cooperate with that logic. It never did. The Cherokee matrilineal system absorbed strangers for centuries. The political confederation called Cherokee made room for people who arrived from outside its original boundaries. The matrilineal clan structure meant that a child's Cherokee identity came through the mother regardless of the father's origin, which means that every Mediterranean trader, every Scottish frontiersman, every person of African descent who had a child with a Cherokee woman contributed a lineage to what would eventually be called Cherokee DNA. The Trail of Tears then imposed a brutal genetic bottleneck. Roughly 4,000 people died on the march. The Eastern Band survivors, perhaps 1,000 people who hid in the Smokies, became a disproportionate genetic reservoir for what we now study as ancestral Cherokee lineages. The removal did not just kill a quarter of the population. It selected which genetic signatures would carry forward. It chose the archive, not the U.S. government, the mountains, the women who ran. The bloodline they tried to police was always a river. Every population that has survived long enough, the Cherokee, the Romani, the Melungeons, the Samaritans, the Parsis, leaves a genetic record that looks like this. Not a clean line, but a document. A palimpsest. Layer on layer. Rewritten, but never fully erased, carrying the traces of every encounter, every adoption, every forced migration, every child born at the intersection of two worlds that history said should not have touched. The Cherokee case is not a curiosity in the margins of Native American genetics. It is a warning about what we think we are asking when we ask where a people came from. Millions of people today are sending saliva samples to labs, hoping for clarity for a clean answer. A single origin, a bloodline that explains them. The Cherokee data suggests that the clean answer is almost always the wrong answer. Complexity is not the failure state. Complexity is the record. The science is not finished. No comprehensive ancient DNA study of pre-contact Cherokee burial sites has been completed at scale. The haplogroup frequencies found by Yates and his team remain contested, their origins unresolved. It will take another generation of research, at minimum, to know whether those anomalous maternal lineages arrived before Columbus or after, and whether the distinction, in the end, changes what we know about who the Cherokee were. What we know is this: they survived the removal. They survived the blood quantum regime. They survived every attempt to define them into extinction. And the DNA survived with them, passed down through the mothers, hidden in the highlands, preserved in the cells of every person who refused to be counted. The Cherokee were never who we thought. And the deeper the data goes, the more that turns out to be true of every people who ever held a mountain long enough to call it home.